Okay, here we go. Defining the central nervous system, we should know that it is comprised by the brain and spinal cord. And these soft tissue neuronal structures are in turn housed by the axial skeleton. And just to remind you, the axial skeleton, the elements is they are the skull, vertebral column, including the sacrum and coccygeal bones, the ribs, the sternum, and that isolated hyoid bone. In any event, when the appendicular skeleton finally evolved and came to be, along with its muscles, what was needed was an extension of the central nervous system to communicate with those muscles that move the appendicular skeleton. And that was in the form of these blue lines that you see before you, the peripheral nervous system. So basically, the peripheral nervous system communicates with the spinal cord, and the spinal cord is the information highway that connects the brain to the upper and lower body. And just to give you some numerical data, they are about 10 to the 14 synaptic connections in the human brain. So the peripheral nervous system has been divided up into a somatic nervous system and the autonomic nervous system, but I want to concentrate for now on the somatic nervous system. Its target, the striated or skeletal muscle, and the nerve fiber type designation to that target is referred to as GSE fibers, or general somatic efferent fibers, because they come from somites. The sensory innervation comes in the form of GSA, general somatic afferents. And that's it. There's only two nerve fiber types that are affiliated with the somatic nervous system. The autonomic nervous system, I will get to that later on, but briefly, the autonomic nervous system is considered an efferent system. It only has one fiber type, the general visceral efferent fibers, general visceral, not somatic, because they don't come from somites. Their embryologic origin follows a different course. And the three targets to the autonomic nervous system is the cardiac muscle, which you don't have to worry about until Unit 5. But the other two, yes, smooth muscles is one target that you should be interested in, because the smooth muscles line the arterial walls of vessels, right? The smooth muscles at the base of the body hairs in the form of the erector pili muscle. That's smooth muscle, so therefore it is under autonomic territory or control. And glands, which we have many. We have the suprarenal glands, we have sweat glands, lacrimal glands. We have a whole bunch of glands. So that falls under autonomic control. So what happens to this guy, the GVAs? Well, they're orphaned. But usually the rule of thumb is wherever you have GVEs, you always have GVAs. These are the sensory innervation of these targets, and usually these visceral targets or structures have a small amount of these fibers compared to the GSA, which has a lot more, and therefore the precision of feeling something is far greater in the somatic than it is in the autonomic. So taking a look at these two typical vertebral structures, one on top of the other, it creates the intervertebral foramen, and that is the exit point through which the 31 pairs of spinal nerves, the spinal nerve emerges through here, but they also emerge in the front and back of the foramina of the sacrum. But here, in the intervertebral foramen, the spinal nerve emerges before it bifurcates into a ventral ramus and a dorsal ramus. looking at the spinal cord as it travels through the vertebral canal, it is encased by three meninges, starting from deep to superficial. You have the more intimate pia mater, then the arachnoid mater, and then the gray here is the dura mater. And now you can see the spinal nerve exiting through the intervertebral foramen, and you can now see the bifurcation of the spinal nerve, which is really a very short segment, and the spinal nerve will give rise to the much larger ventral ramus and the smaller but shorter dorsal ramus as well. You can see the bifurcation very clearly here. By the way, what kind of vertebra is depicted here? And so this is like forcing you to look at the morphological hints that will tell you that this is a typical thoracic vertebra, because you can see the facets and demifacets and full facets on the transverse processes seen here and here on the vertebral bodies, and that serves for the attachment of ribs. Here is a color-coded illustration showing you the entire vertebral column. The blue signifies cervical, the orange the thoracic vertebra, serving for the attachment of the ribs. Then the 5 lumbar, denoted by green, the purple sacrum, and the yellow the coccygeal bony elements. And in this sagittal cut, you can look through the vertebral canal, you can see the intervertebral foramen, and this is the exit point of the spinal nerve seen there. Let's transition now and look at spinal cord regions. This is a very important distinction. Notice that I said spinal cord regions, and the only thing that's in color here is the spinal cord. We're not looking at vertebral regions. We're not looking at vertebral colored regions. We're looking at spinal cord regions. And so pictures tell 1,000 words. So I like to show you, these are the spinal cord regions that corresponds to the vertebral canal. And right away you should see that the spinal cord ends right here between L1 and L2, as you can see there. And so the spinal cord region that corresponds to the cervical kind of matches really nicely the cervical vertebra, but that's about it. After that, when you look at this thoracic spinal cord region, that corresponds from T1 to T10. And then the lumbar region corresponds from T11 to about the start of L1. So you see it's a different region that we're talking about. So when we say there's a vertebral column break at L4, we're talking here, this region, L4. So here's T12, T1, L1, L2, L3, L4. So here's an L4 vertebral break. If I say there is an L4 spinal nerve lesion, then I cannot go to the L4 vertebral level because there is no spinal cord there. So when I say a lesion at the L4 spinal cord region, I have to go to this region here, depicted in this illustration, and the L4 spinal nerve is around here. So not vertebral column. So you have to look at how the question is worded. I had one of your classmates doing a question in the BRS book that specifically refers to a spinal cord lesion, and the confusion lied in noting that the stem of the question was talking about spinal cord region and not vertebral level trauma. Okay, so speaking about matching the spinal cord to the vertebra, I think I showed you this slide before. This is an 8-week-old, and just to give you a picture so you can better appreciate what we're talking about, this is the 8-week-old embryo. You can see how small that is to the reference penny in this picture. And in this stage of development, the spinal cord matches very nicely the entire vertebral column. Okay, but at 24 weeks, and this is what we call a premature, a preemie, right? The spinal cord is not matching the vertebra because the vertebral column is outpacing the growth of the spinal cord exponentially. And so you can see that the end of the spinal cord at 24 weeks is around between S1 and S2. At birth, and you all know a typical picture of birth, the spinal cord ends around L3, okay? And in an adult, that's me there, ends between L1 and L2. So doing a spinal tap, you might think that going to L3 level is safe, but if you're doing it in an infant, you might hit the spinal cord, so you want to do a spinal tap way below that. Yeah, so the medullary cone is the end of the spinal cord, and that is at L1 and L2. That's called the conus medullaris, right? Then the cauda equina is the horse's tail. Let's zoom that in, and you can see that that kind of resembles the tail of a horse, but in this case, I'm using the hair behind the horse's neck, and it kind of nicely matches that there. Okay, so you can see a lot of things here. So the confusion that I've been hearing is that what's in here? Well, definitely there's spinal nerves in here, but there's also dorsal rootlets and ventral rootlets that are merging to form the dorsal roots and ventral roots. But as they're exiting, the spinal nerves are formed at the intervertebral foramen, and those nerves, the dorsal and ventral roots, continue downward in the sacrum, and when they exit, okay, the spinal nerves are within the sacral hiatus, and if they exit through a dorsal foramina, that would be a dorsal ramus. If they exit through a ventral foramina of the sacrum, that would be a ventral ramus. So understand the distinction. Above the sacrum, when we're talking about the vertebra, the nerves that are coming out are the merging of the ventral and dorsal roots, and as they exit, that's the spinal nerve, and then you can actually see the bifurcation right there of a dorsal ramus and a ventral ramus. And of course, the swelling here is the dorsal root ganglion. So I hope that's clear. But again, send me questions, and that's the only way you progress. So this is a typical illustration of the spinal cord, and it's been cut away so you can see the famous H of the spinal cord. Here's ventral, and here are your ventral rootlets. Ventral rootlets then merge to form a ventral root. Conversely, here the dorsal rootlets coming out through the dorsal horn. Here's the ventral horn, here's the dorsal horn. And these dorsal rootlets then merge to form a dorsal root, and then you have your dorsal root ganglion, and the dorsal root and ventral root merge to form the short segment that we call the spinal nerve. The spinal nerve will then bifurcate into a ventral ramus and a dorsal ramus. I hope that's clear. In the dorsal root ganglion, I hope you realize that that is where the cell bodies for sensory afferents live. That's where they live. Let's take a look at one. It's a unipolar. There is no synapse taking place in the dorsal root ganglion because they're unipolar, right? So this is your typical sensory nerve fiber type. And this character was used. I think when I look at that, I always think of that character, so forgive me for showing it. But, a lot of people say, what's the cell body used for? And they say nutritive purposes, right? They nurture and give nutrition to these axons. So, looking at this very carefully, you can see that the cell bodies, and there's many of them, causes the swelling of that dorsal root ganglion. So here's central nervous system, here's peripheral nervous system, right? The electrical impulse, the signal, is always going towards CNS for ultimate interpretation and then behavioral action of some kind, right? So we use the cell body to demarcate some of the things that I like to show you, but the axon of each dorsal root ganglion bifurcates into a central process and a peripheral process. So the central process is the one that's closer to the CNS, and the peripheral process, I only say that because it's nomenclature and it's good to know where you are. For clinical pathologists in neurology, it's a big deal because certain pathologies target the central processes versus so I just say that for you, just to round out your education, really. Here's a beautiful illustration from Netter. I removed all the labels. It's a beautiful picture. Let me just show you what that looks like. Here is those cell bodies. And look how they come into that causes that swelling. And that swelling is your dorsal root ganglion. So now we can define it. A collection of cell bodies outside the central nervous system we call a ganglion. Okay, so that's it. What about inside the central nervous system? Okay, let's go to the ventral horn of that. How do I know this is ventral? How do I know this is anterior? By the dorsal root ganglion. The dorsal root ganglion tells you this is posterior and this is anterior or ventral. So here comes a cell body going to, a bunch of cell bodies and that ventral horn. Similarly here. So a collection of cell bodies within the CNS we refer to as a nucleus, right? As a nucleus. So here are your dorsal rootlets, and on this image here you see the ventral rootlets, and then together they form the spinal nerves. So this is a real life laminectomy. Here are your dorsal rootlets forming the dorsal root, and back here you can kind of make out the ventral rootlets forming a ventral root, and then together, you don't see it, but the spinal nerve is deep to that. So looking at this, let's take a look at the ventral root and ventral rootlets. Here you see it. And the electrical impulse, you should know, a very simple illustration of physiology. The electrical impulse goes from the central nervous system out to the periphery. That's what that arrow signifies. And those are motor efferent fibers. So anything with an efferent after it, the electrical impulse is going away from the central nervous system to the periphery. So in the case of somatic innervation, it's going to striated muscle. In the case of the autonomics, because that's efferent, but the classification is general visceral efferent, not general somatic efferent. It also has to travel away from the central nervous system, targeting the cardiac muscle, the smooth muscle, and the glands of the body. But again, the concept, the signal is going away. Conversely, you have the dorsal root, the signal has to go back to the CNS. So that's the arrow. That's the reasoning for the arrow. A couple of more things, right? Just so you know, when these rootlets exits the central nervous system, you can see that it merges. So in this particular case, the rootlets are merging very quickly here, But in this particular case, the merging is a little bit further away from the CNS. Doesn't really matter. What matters are these are the rootlets, ventral rootlets, and they eventually will merge or coalesce into the ventral root. So ventral rootlets, then ventral root, then short spinal nerve, a short spinal nerve. Because here are the dorsal rootlets, right? Look at the bifurcations there. They're merging, and then finally they coalesce. That's not the right word. They come together and they form the dorsal root with the swelling of the dorsal root ganglion. And now that's a spinal nerve, okay? So that's the important. And then the spinal nerve will bifurcate into a short, smaller dorsal ramus and the larger, longer ventral ramus. Remember, ramus is branch, and that's the difference between these two guys and these two guys. Forgive me. I have to go a little bit more deeper into the difference, and the difference is now going to be illustrated. Okay, so here the sensory fibers travel only through the dorsal root, and the ventral root only efferents travel. So that is a very important distinction. So this is an exclusive highway for efferent fibers, the ventral root. The dorsal root is an exclusive highway for afferent fibers to come back into the central nervous system. But do these two guys ever come together? Yes, they come together at the spinal nerve. And now all bets are off, and these fibers are free to mix. They're free to mix. So a dorsal ramus will have efferent and sensory fibers, and a ventral ramus will have efferent and afferent fibers as well. Let me show you an illustration to show you that. So this is to remind you of the nerve fiber types now. Nerve fiber types, okay? Genosomatic efferent, genosomatic, that's for the somatics. So let's take a look at something like this. So here is an illustration of the intrinsic back muscles. Can you think of one? Of course you can. You dissected one, the erector spinae muscle, comprised of three: spinalis, longissimus, and iliocostalis, right? So those are intrinsic back muscles. Different from the extrinsic, with the intermediate and superficial muscles of the back. How so? Okay, let's do it. The genosomatic efferent that's going to the intrinsic muscles is denoted by blue. Okay, so let's see. So this is going to be here it is, genosomatic. Genosomatic efferent, the cell body, is living in the ventral horn. Yes, it's going to exit and travel along the ventral rootlets, ventral root, spinal nerve, and now it has to make a decision. Will it take a dorsal ramus route or a ventral ramus route? So in the books and in lab, we said that intrinsic back muscles, the efferent fibers, is transmitted through a dorsal ramus. Okay, so here comes the ventral root, spinal nerve, seen in blue, and now spinal nerve will decide that it will take a dorsal ramus pathway, and the somatic nerve innervation will go to the muscle. Great! So far, so good. It's pretty clear. But all muscles have proprioception, sensory proprioception. Is it tightening? Is it loose? Is it being constricted? Is it compressed? Or whatever. Its proprioception is knowing where the muscle is in 3D space, right? So here's the red, signifying the GSA. It's coming from the muscle. It's going to take a dorsal ramus pathway to get to the central nervous system. So from the dorsal ramus, spinal nerve, going through the DRG, because there's no synapse in the DRG, dorsal root, dorsal rootlets, and now getting to the dorsal horn of the spinal cord, where it will synapse in the spinal cord. That's why I always say says that. So you might say, oh yeah, but the book says internal. I'll leave Unit 6 experts to talk to you about that. But I like to say that it enters the central nervous system and synapses in the spinal cord. The details you get in Unit 6. Okay? Okay, so I just showed you a dorsal ramus pathway. I want to now show you a ventral ramus pathway. And here it is, a muscle saying pec major or pec minor. So pec major, the cell body will start in the ventral horn, travel through the ventral rootlets, merging in the ventral root, traveling through the spinal nerve. Here's the bifurcation of the spinal nerve. Here's the blue. We'll take a ventral ramus pathway to pec major. Oh, so it's a ventral ramus pathway. Proprioception fibers going through red will go through a ventral ramus spinal nerve, but now it has to go through a dorsal root because only afferents will travel through the dorsal root to get to the CNS, to the dorsal horn, well, a synapse in the central, somewhere in the spinal cord. Okay. Okay, so I hope it's a continuation of what I just left off. I had to stop the recording. So I'm using a tree analogy to now differentiate between the concept of roots and branches. I had trouble understanding it when I first got exposed to nervous system way back when, at the turn of the century, really. And so I just want to show you what I mean by that. So let's look at a different illustration. And I arbitrarily put in front of the tree anterior ventral ramus and behind, and on the left side, posterior dorsal ramus. I like to use the tree, the trunk of a tree. Everybody understands it's going to have a lot of branches. If you speak Spanish, the word for branches, ramos. Latin is ramus. So you can understand that that is different from roots, because roots are underneath the ground. So here is a root, and it happens to correspond where the ventral ramus, so this would be a ventral root or an anterior root. Here's a dorsal root, but to give it some credibility, I would have to drawing this is the dorsal, I have to draw in, guess what? Yes, the significant swelling of the dorsal root ganglion. And this is the key. Here it is. When you turn the tree sideways, you're mimicking the exit of the spinal nerve through the intervertebral foramen to represent the exit of the spinal nerve on the vertebral column. That's the key. The ground actually signifies the intervertebral foramen. Okay, that's perfect. And then, of course, let's draw in the ventral root. Let's draw in the arrows pointing to the ventral ramus, and therefore the tree trunk. Well, that's the dorsal root with the dorsal root ganglion, and always the dorsal root ganglion sits in the intervertebral foramen. And look at that, the spinal cord with its ventral rootlets going through and merging with the ventral root. Let's follow some of those axons. Okay, I'm sorry, here's the tree trunk that shows you that short segment of the spinal nerve before it bifurcates into a ventral ramus and a dorsal ramus, which isn't labeled. So what I did was I showed the axons traveling from the ventral horn through the rootlets, the ventral root, through the intervertebral foramen, through the spinal nerve, and going through the ventral ramus, presumably going to a superficial or intermediate muscle of some sort. Okay? But whatever muscle it is, it has sensory afferents going back through the ventral ramus spinal nerve. And now it will go back through a dorsal root, rootlets, dorsal horn, and synapse somewhere in the spinal cord. So I hope that helps you to distinguish roots from ramus. Let's turn our attention to the autonomic nervous system. I want to talk to you a little bit about that. So I kind of showed you the cervical enlargements. There's a cervical and lumbar enlargement, and that corresponds to the brachial plexus. And now that you're in lower limb, the lumbar plexus. But I should not have put that slide in there because it's not in keeping with what I want to talk to you about today. But I saw it, so I have to describe it. Let's look at some cross sections of the spinal cord. So let's look at the cervical illustration, and this is a lumbar histological cross section, and you see the typical H, right? You see the typical gray matter of the H, and surrounding it is the white matter. And here you have the dorsal horn and ventral horn, and really nothing in between, right? Similarly, the lumbar, same thing. You have a ventral horn and a dorsal, but nothing in between. But in the thoracic region, you have your ventral horn and dorsal horn, but now something juts out. You have a dorsal horn and a ventral horn, and now we have a lateral horn. All right, that's going to be important, and we're going to talk to you about the autonomic nervous system very briefly. Just 3 or 4 slides, and I'm already tired. The autonomic nervous system. A little review. The targets: glands, smooth muscle, cardiac muscle. So we call when we divide the autonomic nervous system into parasympathetic and sympathetic, and you might remember the flight or fight response, that's sympathetics, and then the parasympathetic is I forget the something, and you rest, sleep and rest or something like you know it. But so let's start off with the peripheral nervous system. So it is the autonomic nervous system is totally a general visceral efferent based system. It is a 2-neuron system. That means there are 2 cell bodies, right? That if you have 2-neuron system, you're going to have a synapse of some kind. So there will be a synapse, and here is the first cell body, the first neuron, And here's the second neuron, and it's kind of a little distinguished by the dashed lines versus a solid line. And the synapse will take place inside a ganglion, right? So it's a two neuron system. It's going to be a synapse. The synapse will take place in some ganglion of some kind. Okay, so far so good. What else? Okay, so the first axon, the primary axon, is highly myelinated. And when you look under a microscope, it has this whitish appearance because of the histological staining, very white. After the synapse, it is not as it is myelinated, but not as myelinated. So it has a grayish appearance. And now we can be a little bit more descriptive and say that this axon is before the ganglion, and this axon is after the ganglion. So we call this preganglionic. This is a cell body of a preganglionic axon. And this is the cell body of a postganglionic. And now certain books changed it to presynaptic, to postsynaptic. The older term, and it's now returning based on recent articles that I've seen, now we're saying preganglionic and postganglionic. So that's the concept. Okay, so it's divided to sympathetic and parasympathetic, and what you see here is the parasympathetic, and we call this the craniosacral outflow. So it has like 4 nerves that contain GVEs in the ocular motor, facial, glossopharyngeal, and vagus. And again, you don't need to know that for Unit 1. And then segment, spinal cord segment. Remember, spinal cord segments, not vertebral column, right? We're talking about spinal cord, and now you understand the difference of what we were talking about previously. S2, S3, S4 for the parasympathetics. Okay, so not bad. Let's go on. So that is called the craniosacral outflow. For the sympathetics, right? The cell, now I can talk to you with the jargon. The cell bodies of the sympathetic nervous, the cell bodies of the preganglionic, the first primary neuron, right, of the 2 neuron system, originates from the thoracolumbar segment. Thoracolumbar. Wow. So here it is. Oh, it starts at T1 and it ends at L2. And let's color code that thoracolumbar segment in blue. Very nice. So the cell bodies of the preganglionic axons live in the central nervous system, it's in the spinal cord. But where? Where does it live? They live in the intermedial lateral cell column, or lateral horn. I thought I saw that, but I may have forgotten it. Oh, no problem. Here it is. Here it is, and that thing jutting out between the dorsal horn and ventral horn is that lateral horn. And here are the cell bodies of the preganglionics. And here are the motor efferents of these GVEs exiting the lateral horn, going through the ventral horn, going through these ventral rootlets. All these ventral rootlets, and now rootlets, and then merging and ventral root, and then hooking up with the dorsal root, and then becoming the spinal nerve before it decides it continues on as a dorsal ventral ramus. Okay, so I hope that cleared things up. Let's bring this back, and now you can appreciate this sequence. The cervical will have no lateral horn, but as soon as it hits the thoracic spinal cord region, all the thoracic spinal cord will have a lateral horn. L1 and L2 will have a lateral horn, but not L3, L4, and L5. Okay, so I hope that helps that. Okay, the cell bodies of postsynaptic axons live either in the sympathetic chain or some place called the pre-aortic ganglion. Pre means in front of the aorta, and the aorta has three specific areas where the ganglions live, and those are the three unpaired vessels that you'll see in Unit 4. But for now, these ganglion live right in front of the aorta and a place called the sympathetic chain. Let's we'll get to that. You saw the sympathetic chain in the thoracic lab. Let's just continue for now. Just for now. Okay? So the sympathetic nervous system, okay? Let's just say that it's going to sweat glands, right? We have sweat glands in the front and we have sweat glands in the back, right? So let's just take a look at the sweat glands for now, in the front. So how does the general visceral efferents get there? They originate from, in this particular case, a T10 spinal thoracic segment, right? It says T for thoracic. Lateral horn of the cell bodies. The axons will travel through the ventral horn, and here the ventral root, spinal nerve, and now it will initially go through the ventral ramus to enter this ganglion, the sympathetic chain ganglion. This is only one segment. The sympathetic chain travels from the base of the skull all the way down to the coccyx. But right now, all you see is one segment, but the chain is extensive from the base of the skull all the way to the coccyx. So how does the preganglionic sympathetic GVE travels? Lateral horn, ventral root, spinal nerve, ventral ramus, and now it enters the sympathetic chain via this connection. And the name of that connection, that's your intervertebral foramen, the name of that connection is called the white ramus communicans. Communicans means a communication. It's getting to the bridge via this pathway. Why is it white ramus? Remember that the preganglionics are highly myelinated. They look whitish under the microscope. So when they looked at these two connections to this bridge, they noticed this is the white communication and this is the gray communication. Okay, so the solid blue line is preganglionic sympathetic GVEs. They travel through the white ramus communicans. Now they're in this ganglion. It will synapse. So now the postganglionic sympathetic GVEs will travel through a gray ramus communicans right at that spinal nerve. And now it has to decide, would it take a dorsal ramus pathway or a ventral ramus pathway? In this particular case, it's targeting sweat glands in the front part of the body, so it will decide to go through a ventral ramus pathway to those sweat glands in the front. Conversely, if the sweat glands are in the back, looking on the contralateral side, you'll see the pathway from the lateral horn, ventral root, spinal nerve. It will go through a white ramus communicans. The preganglionic will synapse with its postganglionic cell body, whose postganglionic fibers will travel through the gray ramus communicans to the spinal nerve through a dorsal ramus. Why dorsal ramus? Because it's going to the back, the sweat glands in the back. It all depends where the target is, and that's where they're going to go through. So this is just one segment. You don't have a concept yet of that sympathetic chain, and hopefully this illustration shows you multiple segments, and now you can see this sympathetic chain. It really travels from the base of the skull all the way down to the coccygeal bones. So the distribution of the sympathetics can get anywhere via that sympathetic chain. Even though the cell bodies of sympathetics live from T1 to L2, the sympathetic chain allows its reach to go to the top of the head to the tip of your toes. So here's one illustration where the solid preganglionic axons will go from lateral horn, ventral root, spinal nerve, go through a ventral ramus, go through a white rama communicans. It will synapse at that level. And like the previous slide, it could exit at that level. But in this particular pathway, in this particular pattern, the pathway after the synapse is that it descends down through the sympathetic chain and will decide to exit through a gray rama communicantes, go to that spinal nerve nexus before deciding going through a ventral ramus. Now, or let's go to the contralateral side, it can enter the sympathetic chain, right? And synapse and the postganglionic can ascend and exit through a gray rama communicans right at the spinal nerve and either go through a ventral ramus or a dorsal ramus. But I do want to bring to your attention a very key point and tap into your great observational skills, because I really believe everyone has great observational skills and you need a little guidance to develop it to a much higher level. So here you see two communicantes, a white ramus and a gray ramus communicantes. You see that there. But when you go up, you only see the gray rama communicantes, and down here, only a gray rama. What happened to the white rama communicantes? It's not here. Well, the white rami communicans only exist between T1 and L2 because that's where the preganglionics are traveling through, creating that appearance of white ramus because of its heavy myelinated axons. So the white rama communicantes is only at the T1 L2 level, and when it synapses at that level and goes be above T1 or below L2, you will not see a white rama communicantes. So you see how you can tell where you are by the presence or absence of a white rama communicans. Now, the people who are crackerjack future neurosurgeons, you're going to say, oh, but this is the sympathetics. Doesn't the sacral region for the parasympathetics also have a lateral horn at S2, S3, S4? And I would have to acknowledge them and say, yes. For S2, S3, S4, they too have a lateral horn protuberance that juts out. But that's for the parasympathetics, and we couldn't show that here because those S2, S3, S4 do not make use of this sympathetic chain. This sympathetic chain is solely there for the assistance of facilitating travel for the sympathetic axons. For the parasympathetic, that's for the ganglion that lives in front of the aorta, the pre-aortic ganglion. Okay, so I don't want to delve too much into it anymore, and I just want to call it, that's it. Okay, so I hope that's helpful. Let me know. Talk soon.